Hello everyone, today's lecture we'll be talking about organic and biochemical syntheses. Mainly we'll be talking a lot about different organic and biochemistry, like I mentioned, syntheses that you'll have to know, as well as some chemical compounds that I think are important for you to know. Okay. Again, this is my introduction with my email. If you ever want to reach out to me with any questions or anything, please feel free. Again, I also highly recommend you join the group. We have a lot of students there. So it's a great place to get a hold of me or the other tutors or some of the other students to get your questions answered, right, or receive any information. All right, learning objectives. What are some organic syntheses you have to know? What are some naming conventions of some complex organic and biochemical molecules? What are kinetic and thermodynamic processes? And we'll actually be talking a little bit about lipids as well towards the end. Okay. First, let's talk about organic syntheses. Starting off, we'll talk about amines and enamines. Many of you will have probably heard of amines, and amines are actually very similar, except instead of having a single bond and two H's, they have a double bond and one H. You can see right here, this is an amine compared to an amine, which would look kind of like this, an NH2, right? So amine, very similar. Enamines, they can be considered like amine derivatives, but really it's easily transitionable between this state, this form and this form, the amine and enamine form. The reason for that is if you actually draw it out fully, this carbon atom has 3 hydrogens on it, like this, right? So what you can do is you can take this double bond, move it here, and then just take one of the hydrogens and move it here, and then you'll get this NH2, the single bond, and then a double bond here on this adjacent carbon. And actually, in addition to this, you can also have, it's not drawn here, but doing this would be the same thing, NH2 here, and then a double bond here, right? These are all essentially equivalent, and they like resonate from one to another, and they're constantly in flux. So both of these forms, or actually all 3 of these forms, exist at the same time. At certain, obviously, levels, right? In different amounts. Okay, so it's very easy to transition between an amine and enamine. And we'll talk a little bit more about this in a minute, but let's talk about how we synthesize amines. Mechanism for amine formation, you start off with a ketone, as you can see here. Then you would have to add in a, you just protonate it with something like HCl, like X usually is for a halide, like a chlorine, bromine, something like that. So you need something acidic like HCl or HBr, right? And just protonate it. Then once it's protonated and you have this positive here, you would just add any amine that you want. Remember, amine is any R group that ends in an NH2. And what this does is this will go into the tetrahedral intermediate. Remember, any double bond O, right, any ketone can go into a tetrahedral intermediate like this, and then let's just put, doesn't matter what it is, R group, right? And the reason it's tetrahedral is because any carbon that has 4 bonds to a substituent is in the tetrahedral geometry, so we call this the tetrahedral intermediate, right? Remember, the way this works is this goes like here, so you get an O negative, and then a C. This C right here is a positive, and then something negative will attack the positive carbon, and it'll form the tetrahedral intermediate, and then this O will get an OH, which will cancel out the negative. Right. So you start off with the ketone, protonate it, right? Add in the NH2, the amine, right? And then you get this tetrahedral intermediate, which will like clean itself up essentially, and it'll resolve into this form, right? Now, you notice that there is now, apart from here, there's water as well as NHR, right? What can happen is the water can act as a leaving group and just leave, be kicked off. It'll go into this iminium, right, intermediate. Once the water kicks off, then you form the double bond with the N, and then simply the H gets kicked off, and you're left with this amine. And then again, remember, amine, and then it can also go into an enamine, right? It's the same thing, enamine, right? Couple of things. This process is fairly very simple. You're essentially just adding, like, if you want to think about it, you start off with this ketone, you put an amine in there, form this tetrahedral. This is the most important. These are the most important steps. Go from here to here, and then you just kick off this OH, and then you're just left with this, where you have to double bond it, and essentially you're just going into here. Those are like the main steps, and then everything in between is just kind of like the cleanup and like the transferring, just to make these big steps possible. But really, these are the three most important steps. Right? Everything else is just essentially cleanup. Okay. Another thing I want to mention: once you do get to this stage, what can also happen is that what we talked about is that the OH gets the hydrogen, becomes H2O, and then acts as the leaving group and just leaves. And that's how you progress to this stage. But what can also happen is this can also act as a leaving group. This can actually leave, and then you just go back to the OH, you make the double bond with the O, and you'll actually just go back to this, right? So sometimes it'll go from this step to this step to this step, and then from here it'll go back to step one, because this will come off as the leaving group instead of the OH. Sometimes that'll happen, and you just go back to the start and then keep retrying. And this will happen when you have a big tube of it. Obviously, a lot of them will do this, but it'll just keep going basically to start. Then it'll go forward again, back to here, then forward again, back to here, forward again, and then eventually it'll go here, and then it'll go to completion. Okay. Just wanted to point that out, but yeah, mainly remember the big steps. Okay, so that's imine formation. Next, we'll talk about the Gabriel synthesis, which is fairly simple, and it's used to convert alkyl halides into amines, which amines, as you actually saw from the previous synthesis, amines are very, very important building blocks used for other reactions. It's very crucial to be able to synthesize amines very consistently, okay, and in the way that you want. So this Gabriel synthesis is a great way to do that. It's very important in many other reactions because it creates these amines. Essentially, you can start off with any alkyl halide, which is, you can have a carbon like this, really big carbon, and at the end you have like a Cl or any other halide. It can be like a Br, fluorine, whatever, doesn't matter. Any kind of halide at the end, and using these two things, which are called, this is called the phthalimide, right? And it's a salt because it has this negative charge, and it also has potassium. So this one's a potassium salt. So this phthalimide salt, and this hydrazine, which is this thing. Hydrazine uses these two as reagents, and you can turn this straight up into this, which is again very useful. Essentially, you just replace the halide with an NH2 group, turning it into amine. And the way this works is very simple. Cl is a very good leaving group, so this the Cl will come off very easily. This will have a partial positive charge because the Cl came off this carbon right here. This nitrogen has a negative charge because it's a salt. It'll attach into the place of this chlorine, like as you can see right here. And then you just do NH2, which is hydrazine, and it just replaces, it comes in and it can, one of these NH2 will replace this, and the other one will kind of get rid of, like basically it'll just get rid of the this bulky group and put an NH2 in there instead. Okay, very easy, very simple. Not much to talk about Gabriel synthesis. Just remember essentially what you can do by putting in these two things. You can turn this into this. Very simple. And if this was an organic chemistry class, it'll be written as this. Essentially, just something like this. It'll have this thing. I'll just put it over here because I don't want to draw it. And then NH2. Two of these, right? And then what is this? It makes X, right? And then you have to just determine what X is, and it'll be like this. Okay. And that's something like you may see on MCAT, like what kind of product will you make if you do this reaction, right? And the product would be like this, something like that. Again, very simple. Next, this one's a little bit more complicated, but also very important, Strecker synthesis. And this one's a little bit more biochemical than organic chemistry, although it does use a lot of organic chemistry. And this synthesis is used to convert an aldehyde into an amino acid using a cyanide and ammonia as reagents. Okay. So starting off, as I mentioned, with an aldehyde, right? Aldehyde has an R group on one end, an H group on the other end, and then a double bond O. Okay. Firstly, you add in a ammonia, right? We're using ammonia as a reagent. You add in ammonia. Again, make your tetrahedral intermediate. That's always essentially, if you see a carbonyl carbon with a C double bond O, and there's organic chemistry involved, most of the time you should assume that at least one of the first two steps is you're going to a carbonyl carbon or tetrahedral carbon or tetrahedral intermediate, right? So again, you're going to your tetrahedral intermediate. You have the oxygen right here, nitrogen right here. Again, clean up into proton transfer. This is your final tetrahedral intermediate. The OH takes the hydrogen, becomes H2O, comes off. You have your, this is your, remember, looks very familiar. This is your imine. So you have your imine intermediate. Then from there, you have, now you're using cyanide, right? Some kind of cyanide derivative, right? And it releases cyanide, like a cyano salt or something like that. This replaces the, this comes in again, reforming the tetrahedral intermediate. Essentially, this is all like very detailed. Really, what you have to know, I would say, is that when you add an ammonia and CN, this one, the CN, the cyanide, to a aldehyde, you just go from here to here. Like this is all extra stuff, like to know the exact mechanism, but really this is the important part. That when you add these two reagents, like this, NH3 and CN, essentially this is what you have to know. You get this part. You replace this double bond O with an NH2 and a C triple bond N. And again, this is very similar to imine formation, is also very similar to a lot of other mechanisms that you probably know, forming the tetrahedral intermediate and getting rid of one part. And again, remember from here, it can go back to here and then just cycle and cycle and cycle through over and over again until it finally goes to completion. If, for example, instead of the OH getting kicked off, the NH2 gets kicked off. Again, that's important to remember, but yeah. So moving on to the next step, now we have, because again, this is still not an amino acid. You know what amino acid is, but this is not it. Starting off here, protonate it. Again, putting in a lot of these H2O, whatever, and then you get this intermediate. And again, here, again, I would say that the important thing to know is probably this. Again, I would say the important thing to know is probably this. This is all like filler stuff because it's like more mechanism, and it is nice to know the mechanism, but especially for this mechanism, it's a little bit complicated. You probably aren't going to be asked to know the mechanism, but you might be asked to know the starting product, the end product, and reagents, right? Again, you use protonation, water, and ammonia, right? And you can turn this essentially into this. Instead of a CN, you turn that into this, right? And everything else is the same. But here's the mechanism if you are interested. I'll go over it very quickly. This the electrons from this bond go into this to neutralize the positive charge. This ends up getting a charge, a positive charge. So that means it is now allowed to bond to this oxygen from H2O. Sorry about that. Then the nitrogen can take the H from here, thus reducing the positive charge on O into neutral. This takes a positive charge and can redistribute the OH tags here. This can form another tetrahedral intermediate right here, right? Tetrahedral. Now you have two tetrahedral intermediates. This one, as well as this one, right? You have two tetrahedrals stacked on top of each other, essentially. Again, proton transfer, you can take this comes off, right? This ammonia comes off, and this goes in to make a double bond here. Okay. And another important thing that I do want to actually stress, because I did go over this, when you're going through an organic chemistry synthesis, usually with reagents, whatever you put in, whatever you put in, you have to put out. So for example, we added in the start, we added ammonia, but in the final product, we don't really have it here anymore. So we kind of have to take it out, right? We usually have to take it out. We put in hydrogen H plus protons here from the solution, so we have to take H plus protons out. Okay. Next, again, like I mentioned, the important steps are like this that you probably should know. Okay, rather than the mechanism itself. But if you'd like to, please feel free. Okay, next, this one's a little bit more important: keto-enol tautomerization. Similar to amines and enamines, this process between ketones and enols can happen spontaneously, and spontaneously, although it does happen very slowly, and it uses an intermediate called enolate, which looks like this. There's actually two enolates, again, resonance forms, this one and this one. And again, if you just move this here, you have this one because, right, it makes sense. So you can easily transfer between the two. Ketones, enolates, again, if you notice, it's the same thing. Essentially, to go from the enolate to the enol and the ketone, you would you put a positive onto here, if you put a hydrogen onto here, right here, you'd neutralize this negative charge. You just have a regular ketone. If you have this enolate intermediate, you put a hydrogen onto here, neutralize this, and now you just have an enol. Okay. So the enol is just, you have a double bond next to this OH. Okay. So let's go a little bit into enolate chemistry, because enolate chemistry is actually kind of important. There's a lot of stuff you can do with enolates that you can't otherwise do. For example, if you were to start, and again, because enolate is the intermediate of going from a ketone to an enol, you can actually turn a ketone like this into an enolate, right? This is the intermediate. And now, starting off with a ketone, which is a very common like starting reagent that we use, is just a ketone. Then you just want to add something to it, or you want to do some chemistry with it. The easiest way is, one of the ways you can do that is through, like we talked about before, forming the tetrahedral intermediate and going that route. But another way is doing this enolate, right? And if you want to add, for example, onto the chain. So as you can see here, you have this enolate, right? This ketone and this enolate. You wanted to add X, which is again a halogen like chlorine, fluorine, bromine, something like that. You wanted to add that to right here, where you do that. You, from the enolate, you could just put it in, and then you would add it to the end. You can add it to the end. You can add another R group, for example, to the end. You could add, in this case, you can add another ketone, right? This is another ketone. You can add another ketone, but only on the carbonyl carbon. And this is important because you notice here, this gets attached by this point. So essentially, you're kind of sticking them, like if you were to think of it like a chain going this way and then a vertical chain, you're just doing, instead of doing this, you're doing this kind of, right? You're sticking them like in perpendicularly to each other, right? By the carbonyl carbon of this ketone, not the enolate ketone, but the ketone that you're adding in. Okay, so if you start off with a ketone, turn it into an enolate, then you want to add in another ketone to the end of the enolate, right? To the end right here into this spot, you will add the carbonyl carbon of the ketone that you're adding. Okay, so this is the end of the original ketone. in black. The blue is the ketone that you're adding, right? And it adds. So if this was, for example, going horizontally, this was going vertically, instead of doing end to end, you just stick it on, right, perpendicularly by the carbonyl carbon. Again, that's important because in this one, right, in this one, now you're adding a ketone on end to end. In this way, it's a different way. This time, instead of attaching by the carbonyl carbon, you have to introduce a double bond, right? Double bond right here, and then you end up attaching by the double bond, like this. So the double bond used to be here. So instead of the double bond, you break the double bond and attach to here, like that. Okay, hopefully that makes sense. And the reason for that is you can just add a hydrogen. I'm not going to go into the mechanism because these mechanisms are pretty long and very kind of, I would say, unnecessary to know. They're a little bit extra, but the actual processes and the reagents they use are important, as well as the products you make. So again, you can just add a hydrogen onto here, right? Because if you cross out double bond, this would be positive, this would be positive. You need to put in a hydrogen to make this, and instead of putting in another hydrogen onto this one, you're attaching it to the carbon, right, on this end, to fill in that bond slot that was left open. So again, if you want to add a ketone to another ketone, if you want to add perpendicularly, just add it straight to the enolate like this. If you want to add it end to end, you have to do it this way with a double bond. You can't do it by the carbonyl carbon. Okay. And those are two important things. Next, let's talk about kinetic versus thermodynamic enolates. As mentioned, enolates are very important as they allow you to easily add groups onto a ketone or an enol. Due to resonance structures, there are multiple ways to make an enolate and actually different products that may come out. These two products are called the thermodynamic and the kinetic product. Again, if you look at it, these two are resonance forms, and they're both enols, right? They're both enols, and they can both be turned into ketones if you wanted to, but shown here is the enol form, right? And just like you would expect, you can turn these enols into enolates, right? But if you look at these two enolates, they can actually turn into a different enolate. They can turn into different enolates. That's important because now they have a negative charge on different spots. And remember, the thing that you're adding onto the enolate, it always adds on to where the negative charge is. So for example, if I wanted to add Cl2, for example, if I wanted to add a chlorine group onto these, if I was to be adding it onto this one, I would get this as my product. Whereas if I wanted to add it to this one, I would get, right? You can see how this and this are different molecules, right? Even though you start off with like kind of like the same thing, based on the enolates changing, you can actually get different products. What this is called, it's called the thermodynamic and kinetic enolate. And actually, I'll cover it in a second on the next slide. And again, here you can see this. You add in kind of the same thing, and then you get different products. You can get this kinetic product, or you can get this thermodynamic product. Obviously, they're not the same. The general rule is adding to the most substituted carbon is the thermodynamic product, while the other one is the kinetic product. I'm not going to go fully in depth as to exact differences, what makes something a kinetic product, because that's, I think, very much beyond the scope of the MCAT. But the basic idea is it's very easy to determine which the thermodynamic product. The way you determine that is this negative charge. This negative charge, when you put it onto here and then substitute it like that, you get this. And here, the negative charge is on this carbon. This is a tertiary carbon because it's bound to 1, 2, 3 carbons. So it has 3 carbons and 1 negative charge. What that does is having tertiary carbon or a carbon that's bound to 3 other carbons and has negative charge, it kind of, the other carbons stabilize the negative charge a little bit. Whereas with this one, you have here, you have the negative charge on this one, and this is called a secondary carbanion. The reason it's called a secondary is because it's 1, 2. This carbon is only attached to 2 other carbons rather than 3. Because of that, 2 other carbons don't stabilize the negative charge as well as 3 carbons would. So thermodynamically, it's more favorable to have the tertiary carbon. And so that's the thermodynamic product. However, the kinetic product, I won't go into it why it's the kinetic product, but essentially, if it's not the thermodynamic product, it's the kinetic product. And that's a very safe assumption for the MCAT at least, right? And it's very easy to determine which is the thermodynamic product, whichever is more substituted, which is always going to be a tertiary over a secondary, or even a secondary over a primary. And a primary would be like an end carbon, for example, look like this. This carbon right here would be a primary carbon because it's only attached to 1 other carbon. Okay, so it'd be like on the end, would be an end carbon, would be primary. Okay, and as you can see here, the temperatures, right, in the situations that you would use to add this ethane, right, because you're adding an ethane to both of these, the temperatures to add kinetic thermodynamic product for are different, right? Because thermodynamic product, you can do it at a higher temperature. This is like just normal room temperature, 25 degrees Celsius. Whereas obviously for the kinetic product, you need to make it very, 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 very cold, right? Because you need to offset some of that like thermodynamic unfavorability, right? And make sure it goes only by kinetic rules. If you do it at any higher of a temperature, it won't do the kinetic product. It'll default to this. Okay. Hopefully that makes sense. Remember, kinetic product is one that's less substituted. Thermodynamic product is one that's more substituted. So tertiary is the best, secondary, and then primary. For a thermodynamic product, you can do it at room temperature, whereas a kinetic product, you must do it at very cold temperatures. Okay, next let's do a little quick talk about lipids, especially biochemical lipids that we commonly use. First, let's talk about sphingolipids. So before, when we talk about lipids, we're generally talking about triglycerides. Those are those main lipids that we use to metabolize, right? Fats, when you think of fats, that is triglycerides. And these are the ones that are used in metabolism mainly. There are other types of lipids, actually multiple other types. The first one is sphingolipids. These are usually found on cell surfaces. They're used for signaling, recognition, the actual structure physically. The name sphingolipids derives from a unique backbone molecule that is found in all sphingolipids called sphingosine. And as you can see, this is what sphingosine looks like a regular fatty acid, except it has this unique backbone, which looks like this, essentially, this part. This part is essentially what makes a sphingolipid a sphingolipid. Here in the bottom right, I actually put it in contrast. This is a triglyceride. Triglycerides have glycerol. That's why it's called, or diglyceride or monoglyceride. It would have glycerol attached to it, to its backbone. Whereas sphingolipids have instead this like weird thing instead of a glycerol. It's a little more complicated thing, but yeah. And then you can see this has R group. If there's R groups in H, it's called ceramide. Phosphocholines called sphingomyelins, sugar, whatever. There are different names. You don't have to know this. If you want to, you can, but it's not important. Main thing, sphingosine looks like this. If there is a sphingosine in a lipid, it's a sphingolipid. Very simple, right? And that's compared to a glycerol, which looks like this, which would make something a glyceride. Okay. And sphingosines are not usually metabolized. They're usually used for cell signaling and similar to a kind of like a phospholipid bilayer, right? Phospholipids are also a different class of lipids, but similar to phospholipids, sphingolipids are mostly for like cell signaling and recognition between cells. Another type of lipid that certain animals have in their bodies is called wax. Wax are also lipids, and they have an ester linkage connecting the chains instead of a glycerol or sphingosine backbone. Many plants have wax on their leaves, which prevents water loss and controls evaporation, just so the leaves don't dry out or get too wet, absorb too much water. Bees and other insects also commonly produce wax. That's why it's called beeswax. It produces wax part of the construction of their hives for like honeycombs, a lot of beeswax, right? It keeps like structural integrity. They look kind of like this, right? As you can see here, this has a bit of a unique backbone, which kind of looks like this. It's an ester instead of, again, a glycerol or sphingolipid. This one has an ester, which is a double bond O attached to a ether or a connecting like O, right? This on its own is called an ether, but when you have a double bond O here as well, it's called an ester. Okay. Another type of lipid that we want to talk about are called fat-soluble vitamins. These are actually very, very common in biology. You've probably taken some very recently as well. Fat-soluble vitamins either in your food or as supplemental tablets. These are very common vitamins, and they're called fat-soluble, and they're considered lipids, right? Because remember, if it's a fat, it's dissolved in fat, like dissolves like. There's water-soluble vitamins, which are dissolved in water, right? Because they're more polar. Vitamins A, D, E, and K are fat-soluble, whereas all of the vitamins B, so there's a bunch of vitamin Bs, all of the vitamin Bs and the vitamin C, those are water-soluble, they're not fat-soluble. Vitamins A, D, E, and K are fat-soluble. I'm not going to go into what all the vitamins do because they do thousands of things, and each one of them does different things. You don't really have to know all of the specifics of what each vitamin does. If it ever comes up, it'll come up in a passage on the MCAT, and they'll tell you what each vitamin does. But you do have to know that they are fat-soluble. They say vitamin A, for example, or vitamin E. They play a lot of roles, and an important thing is that they tend to stay in the body for a long time. They're very hard to get rid of. Because of that, these types of vitamins can actually be very toxic if ingested in excess. That's why it's very, very, very bad to take a lot of vitamin A or vitamin K tablets. Those are very highly controlled, especially like some kids might, like little kids, obviously they don't know any better. They might take a lot of vitamins A's and they can get poisoned actually off that. It can lead to things like blindness or vomiting or possibly sometimes even death. Take too many. That's a trait of only fat-soluble vitamins. So for example, water-soluble vitamins like vitamin B and vitamin C, you can have pretty much an unlimited amount and it won't kill you, won't do anything to you, but fat-soluble are toxic at high levels. So yeah, just remember that. This is what vitamin A looks like. Right, again, just a regular lipid, but it has this, it's kind of like backbone. Essentially, instead of glycerol, sphingosine, or an ester, it has this kind of group. Okay. And I think, yeah, this is the last type of lipid that we want to talk about. It's called a prostaglandin. These are very small molecules, and they're released by actually pretty much almost all the cell types in your body can release prostaglandins. And they're released in response to injury or infection, and they begin the process of inflammation and clotting. Whenever you get injured, you get inflammation in that area, and you also get clotting in that area. That's started by these molecules called prostaglandins. Okay, again, always for lipids, the main thing to look out for, and the main thing that'll usually be different is this part, backbone. This is what one for this one, alprostadil, which is E1 in the cycle. I don't believe you really have to know too in depth the inflammation process, but anti-inflammatories usually, when you take like a non-steroidal anti-inflammatory drug like Motrin, ibuprofen, whatever Tylenol, they usually will target the production and like the kind of like the process of prostaglandins. So that this is kind of like what starts the whole inflammation and clotting process when you get injured or sick. Okay. And they're also lipids, of course. Okay, so hopefully that was helpful. Just to get a little bit more information about lipids, as well as some important syntheses and like organic chemistry stuff that you may have forgotten or wasn't covered in the previous lectures. Hopefully this helped. If you have any questions again, please feel free to email me. Otherwise, thank you very much for your time and have a great night.